So welcome back, everyone. We are now moving into the final session where we tried to integrate a lot of the different themes and information from the whole workshop. So we're going to first have Dr. Chirag Patel from Harvard University do a general summary and some key takeaways of what was covered in the 4 sessions from the last 2 days. And then we're going to have Dr. Carrie Nadeau from Harvard University that will moderate a final open discussion. So we would like to come away from this final closing session with some key concrete recommendations for moving this field forward. And we do encourage anyone that has any final comments or suggestions or questions that didn't get covered in any of the previous sessions to please speak up or enter their comments, questions into the Q&A box in that final discussion. So I'm going to now hand it over to Chirag, who admittedly we gave a pretty daunting task to try to summarize what we've talked about in the last 2 days.
Wow, it was amazing 2 days. Thank you to Kim, Leah, and all for organizing this workshop. I've learned so much, and I hope I do all of you speakers and participants justice in this summary and to provoke discussion, which I'm very much looking forward to at the end of this, and that Kari will help moderate. All right, so I begin with a very strong statement here. As has been set the stage, and as the preliminary data that have been emerging from this workshop, I'd like to say that potentially exposures are causal for cancer. They must induce a biological change in some sort of response or some sort of phenotypic response that could be potentially could be measured. And I'm going to put the question out there, can this biology be interrogated with multi-omic data? And I think what I've learned from this workshop that certainly it can. And certainly we've had amazing preliminary data to suggest that it can as well. So here's a great paper that was called out by Kim at the beginning of the workshop from Drs. Wu, Eckhart, and Baccarelli, who also moderated a session, to suggest and show multiple publications in which interrogation of the exposome with respect to multi-omic responses is in fact possible and could help us get to the ways that the exposures may induce phenotypic change and ultimately clinical outcomes, which we all care about. And Drs. Archer and Ellison set the stage in themes that have been recapitulated throughout the workshop. And that, first of all, the exposome or the environment is complex. There's a heterogeneity of measure, heterogeneity of unit of analyses, and heterogeneity, of course, during the life course, time of day, location, and who we are. I would also posit that, and also another theme that has emerged, that the environment, genetics, and clinical outcome is strongly connected to demographic structure, be it ancestry, race, where we live, or sociodemographic way of life. And also what I've come to respect as well is that cancer etiology is very complex. And complexity of cancer, like other complex diseases, it could be informed by the high throughput study of environment and omics. And so why cancer? There's hundreds of types. There's diversity within an organ. As we've learned, there's risk windows and temporality of both exposure and phenotype, and timing that it becomes a very challenging task, hopefully tractable by the resources that are made available nowadays, like what we'll discuss today. But there's lots of phenotypic variation to be explained. So twin studies or lack of penetrance of genetic factors have led to potentially the role of environmental factors, of course, and there's been established environmental factors with cancer. And another important thing is that there's disparities in both clinical and biological outcomes. There's this confluence of not only who we are, but of our medical decision-making, screening, and therapeutics that have been advanced in cancer. Okay, and so there have been established environmental factors. So outside of this morass of the exposome, we have great evidence to suggest that viruses, bacteria, and pollutants in smoking are connected with cancer in some way. And through history, epidemiological history, we required new studies and associational models to study this. So for example, you think back of Dahl and Hill, back in the 50s to the 60s, positing smoking and lung cancer mortality and later cardiovascular mortality, later leading to a heuristic for this thing called the Bradford-Hill criteria, which we all have in the back of our minds when we're looking at our data, but could be maybe relevant or maybe not so relevant in the multi-omic era. So we talked a little bit about association sizes, how everything is correlated with everything else in the exposome. We talked about replication and the resources that are brought to bear from NCI and others to see how our signals compare across different cohorts. Talked a little bit about specificity and the ways that it might be important in looking at things like Mendelian randomization. Temporality was a major theme throughout the conference. And of course, questioning dose response and linear dose response and potentially and non-linear dose response as brought up in our last session and in our integration session. And we have biological models that have been posited as well. And so how does multi-omics play into all these models, I think, is a question that we need to answer and has been touched on in our third session. So of course, we've had the multi-hit model for tumor suppression. I learned about the tumor promotion hypotheses. And of course, there's this provocative statement about random and stochastic nature of stem cell divisions that is also correlated with prevalence. And so how do we use multi-omics to partition out what is truly stochastic from those that are driven by genetics and environment? So a new paradigm of how to select exposures is another possibility. So exposome-wide association studies for discovery at E. And it's become clear in this workshop and others that have been considering the exposome that it's quickly becoming possible to measure thousands of analytes that could include endogenous action by our bodies, but also exogenous exposures potentially at scale. And as has been accelerated the GWAS community towards high throughput associations, genetic variants and outcomes, can we in the exposome community achieve this cost efficiency with exposome markers? And so measurement of these analytes and geospatial markers are soon to be commoditized. And so can we expect this GWAS level of efficiency from the exposome? So the first session, as all of them were very important, but this one was great to set the stage about cancer susceptibility, environmental exposures, the importance and eminence of study design, and disentangling etiological pathways for candidate exposures and clinical risk factors for cancer risk. And so why study design matters, and we start with this very important statistic of prevalence and incidence of cancer. And you see that disparities persist even though mortality is decreasing for all groups. And you have different methods in your cohort designs in your armamentarium to traverse inference and generalizability. So what you use for your specific question, I think, was a major discussion point that was brought up. And approaches such as targeted sampling may increase efficiency. So upsampling individuals who are at high risk or predisposed. And a very important topic that was brought up throughout the workshop is that different types of cancer will have different windows of susceptibility. And so development of key tissues, such as the breast, is exemplary for identification of these windows. You think of, as someone develops from their fetus onwards, how do we identify what those key windows are, and how do we map that to outcomes? It seems like an intractable task, but as exposures and the multi-omic changes through this life course as well. But I think there's an opportunity here for mapping these exposures in multi-omic science by potentially stitching together different data sets, different cohorts, as was being brought up throughout the workshop as well. We learned about Mendelian randomization, a way of using or harnessing genetic and environmental correlation, this wonky idea that genes could control what we're exposed to potentially, and using that as a natural experiment to move outside of the world of confounding and reverse causality as an instrument to map towards a clinical outcome. So you avoid potentially confounding of things that could be confounding your multi-omic or exposome assay by looking at the genetics that predispose one to that particular exposure. These, I think, data could be easily scaled. So we have GWAS is exploding, as was mentioned. There's many opportunities to do Mendelian randomization across the phenotype spectrum of cancer. As of today or a few days ago, there have been 471,000 associations been published and publicly available in the GWAS catalog. And one outstanding question is, can we develop an exposome catalog for exposomic studies? And push button analyses of these studies are available as well. So things like MR-base are available to input a GWAS data set, input a clinical outcomes from a GWAS summary statistics, and outcomes your causal analyses. So we had a fantastic discussion in this cancer-specific discussion about, of course, the preeminence of study design and linkage to the sample matrix. So you could be looking at tissue, you could be looking at dust. So what sample types should be examined? How do we coordinate the timing of measure of exposure and of the phenotype? We talk about variant versus genetic versus biological susceptibility. How do we assess this, and what windows to capture this time dependence of total biological susceptibility? What are efficient designs, case controls? You recruit those that are higher at risk or known etiological outcome, but how do you balance that with discovery? And given our knowledge about main effects, what are the power requirements for doing these types of studies? There's a tension versus opportunity of the life course. So as we said, there's these things that these exposures and multi-omics that change over time. So we need to potentially become more efficient and stitch these data sets together to create these opportunities. We need to be fair, have our data sets findable, accessible, interoperable, reusable, and there are efforts from NIH to help us do this as investigators, but align. We need to align those approaches to our statistical analysis and harmonization procedures with practical scientific goals. And there's new measurements, ways of measuring the environment or the exposome. So how do we capture it in the right exposure at the right time and develop these tools to integrate across existing cohorts? In session 2, we talked about computational approaches, so statistical approaches for integrating omics data. A great talk by Dr. Gotterman about the basics of data substrates that we have to do these mapping, and then a real world example that looks at a natural disaster of Hurricane Harvey, looking at these things that could happen acute and chronically over time. So this fundamental thing is you have an exposure, you have biomarkers that might be have induced change due to exposure, and then mapping that to outcome is central thematically throughout the workshop. And so you have different models of consideration. So as a combination of a genetic factor and environmental factor, increase or decrease your risk versus them additively. You have this mediation idea. So this figure shown on the right here that I borrowed from Dr. Gotterman. You have potentially new machine learning methods to look at these networks, and what becomes a challenge is how do you interpret these networks, and things like canonical correlation off-the-shelf tools to look at these correlated data structures in concert. So the challenges that have existed through time in this space include a power and hypothesis space. So multiple discussion questions about what is the number of elements that we're studying? What is the multiple hypothesis threshold of the exposome? There's a vast sea of correlations. So how do you detect your signal from the noise? We have methods such as false discovery rate and p-value thresholds, but how do you balance that with power issues? And complex inputs have complex outputs. So how do you translate these complex outputs to your next experimental step that we learned about in session 3 or session 4? I think there's substrate there for us to examine all of these questions and fill these gaps. So there are cohorts that are out there of multi-ancestry backgrounds with geographic distributions and biosamples, I believe, that are waiting to be analyzed. But things are not so straightforward in mapping exposure. So here's an example that we did where we took a look at genetics and environment on the geospatial level. And I tell you that it wasn't that easy to map a zip code to different exposures that you may want to look at, be it social deprivation or temperature or air quality. These are publicly available, but we still need methods to incorporate these in sensitive data settings. Of course, there have been advances in this area, like use of the DCOS tool, which is a decentralized way of geocoding individuals, mapping them to these geomarkers of exposure to then analyze in specific data contexts or your data warehouse. Amazing convenient sample data sets that have been linked to cancer registries like the UK Biobank with image data, genetic data, exposure data, omics data. But also what we have to deal with when harmonizing these cohorts is understanding the inferential quality of where we can generalize, where we cannot. Of course, biobank samples are convenience. They're not the same as individuals from the general population, as Fry and others show that you have a different population that you're studying in the UK Biobank versus those that are collected in the general population. But of course, there's the All of Us study to help us get there in the United States. Of course, also with genetic data, upsample individuals who are not currently well represented in studies. And I think there's an amazing opportunity to link our gold standard registries with All of Us to look at cancer outcomes, as well as enhancing existing resources. So look at TCGA. Let's take an example of this developing a cancer atlas. Thousands of cross-omics papers that have been supported by Cancer Genome Atlas, the Cancer Genome Atlas program. I think from my outsider view, it's been a tremendous success. And so how can we get exposures into a resource like this to study both genetics and environment with respect to biology of cancer? So data harmonization is something that persisted throughout the workshop, and we're dealing with heterogeneous sources, and to unlock them, they have to be married to hypotheses, as was very well stated by Dr. Kraft. So we have individual level to geographical social determinants of health on the zip code versus what is measured on the individual level. And even on the individual levels, these are difficult to assess, even though there's instruments like Phoenix to help us do that. There have other consortiums out there like PRIME to harmonize polygenic risk scores, and of course, instruments for developing harmonization tools. And one of them is we've been encouraging since 2017 is essentially building a catalog or this index of exposures across multiple data sources, and try to assimilate all this information across to develop a baseline across different populations. And there have been efforts to help us get there in terms of the next level after developing the instruments of helping us harmonize with knowledge-based resources, ontologies. I think I give a shout out to the Environmental Health Language Collaborative who's trying to do this, and funding opportunities also to push these standards out, and as well as technology development for omics research across populations. And so in session 2, we discussed matching the data science to the analytic method and biological hypothesis, be fair, but be aligned. We need to have publicly available data supported by data science resources. Those knowledge bases need to be developed and compute power available to all. And there are great examples from NHGRI to do this. Integration across multiple modalities. I think paring down the hypothesis space and keeping ourselves open to discovery is a challenge. We have thousands of omic factors being measured, so you multiply all this together, it's a data explosion. Approaches to incorporate time, leveraging GEAD by environment correlation to do Mendelian randomization studies at scale. I think it's possible. Triangulation across data. So starting large and with a focus to develop a focused hypothesis, potentially in a large publicly available data set, maybe drive it down to a smaller cohort, and then later on try and investigate it in a biological system. Multiple types of exposure data, very hard to integrate those. Methods to precisely phenotype cancer. Yeah, we have these administrative and registry data. We have ICD codes that are emerging from electronic health records. How do we map that with gold standard outcomes that doctors are actually writing down? There's new ways of also measuring pathology. Ongoing assessment of sources of bias, study design and confounding, and of course, assessment of ancestry, race, and social determinants. All these things are correlated with one to another. How do you develop the methods to untangle this collision of factors? We moved to session 3 and we talked about dose versus burden in exposome research. So this concept of the exposome was made practicable in this next session. And we had examples of omics integration, kind of solidifying some of the things that James talked about with Dr. Marston's talk. And so the exposome is reducing to practice the detection of thousands of chemicals and biological tissue and attempting to map this to geospatial markers to perform exposome-wide association studies. So new platforms for measure, are there fair data available? There are often, or there are ways, emerging ways of mapping to model systems to highlight things that interact with one another, like the microbiome metabolome axes. And there have been amazing examples in cardiometabolic disease, and as Dean pointed out, in liver diseases, and emerging in cancer, as has been pointed out in our discussion. We talked about dose versus burden. moving from something of acute dose potentially to a life course burden, and how to maximally spend given the complexity of exposome, how do you maximize your opportunity given the complexity of what you try to measure. Sort of recapitulating in a figure showed by James is this figure here that Dr. Marsit showed, Carmen showed, about triangulating across different omic measurements to figure out the competing or the interaction between metabolites and exposures and outcomes. Key questions that came are: what is the target tissue? Time is both a bug and a feature. Key clinical questions: what are the key clinical questions to demonstrate utility? And a great example that Dr. London showed us to do this in looking at mapping the exposome to internal omic measurements like the metabolome in pregnancy. This example caught my eyes. I am mostly a cardiometabolic researcher, and this one floored me when I saw this from Shah and colleagues. Is they looked at metabolic signatures connected with diet, so self-reported diet, and used those metabolic signatures to associate with disease. You could just as easily do this in cancer, for example. And so what they found was that these metabolic signatures were more strongly associated to risk than they were on the recall measures that they were actually trained. So what else are these metabolites picking up other than the actual nutrient intake, potentially behavior and others, to get to that response? And so in the integration panel, I think a main theme here was talking about collaboration and participation across multiple researchers and potentially participants together. Also calling for training. This is due to the heterogeneous nature of the data and the studies that need to be executed. I think there's also this need for precision nomenclature. So what is the exposome? How do we communicate what environmental factors are? What is EWAS? There's a confusion in the field about epigenome-wide association studies versus exposome-wide association studies. Or what is XWAS? I saw that flashed up on the screen. What are epigenetics? There's a confusion in the community about epigenetics being falsely just connected with environmental exposure as a one-to-one mapping. And then we have different ways of measuring epigenetics. We have histone marks, we have Illumina arrays, we have RBS, bisulfite sequencing. So we need definitions to help us communicate, measure, and do reproducible science. Let's talk about reference panel for the exposome. What's the baseline distribution? What's the hypothesis space when you multiply all these domains together, these dimensions? I think another aspect of social determinants data is moving from the covariate that we do the adjustment to a criteria for inclusion. So not as a confounder, but something that we wholeheartedly use as inclusion factors. And we need to treat those data in a model-driven way to really tease out their relationship with disease outcomes. I think an important topic is actionability. So you're doing all these omics things, like a polygenic risk score, exposome risk score. How is it going to really impact a person's life? And so what can be modified towards precision health in this way, or precision public health? I think there's a technology gap too. So there were talked about lack of data standards that are on par with genomic standards. So we talked about a little bit about this earlier, but potentially there's these multi-omic measurements, if not causal for the outcome, they could provide biomarkers for for exposure. So potential for cohort portability and potentially indicators of historical exposure that that others like Dr. Hoyo mentioned as could be markers of aggregate exposure, or that example about diet, about historical exposure. And then we move to session 4, albeit I was feverishly working on these slides, so I don't have the great coverage as on these topics, but we had Dr. Hill and Deval reminding us the importance of consideration of the models that we're testing, as well as a great discussion. And so it's possible to bring epidemiology to the lab, as borrowing terms from Dr. Colsino. So we learned about tumor promotion, a model to be tested in non-smoking lung cancer, integration of these ecological data to drive a key experiment with pre-existing mutations in this model system. I think that's the key thing there. And the key in the second was to develop these organoids to test with exposures writ large and seeing how they map to other omic associations. And so you have a potential of doing gene environment in a dish, different tissues, tissue specific to get to the causal connection. Looking at gene environment regulation, this idea of allele specific regulation, very important idea to look at gene environment interaction. Cancer models, we need standards for creation to get to potentially tissue specificity of pathways with respect to exposure. I think there's a great opportunity there. New technology and systems, it was a major theme in this. So most of environmental research is observational, and bringing this to the bench would bring us to causality potentially. And I think a major theme was mapping between the model and the human. So how to model heterogeneity of the human exposome, genome, experience, ancestry in a dish, or more practically, what exposure should we testing, pressure testing first. So I end with my original hypothesis to you is that if exposures are causal, they must induce some biological change, and potentially this can be interrogated with multi-omic data. So thanks to all of you, and looking forward to the discussion. If you like the slides or whatever, please shout out.
Thank you. That was excellent, Shrag. Leah and Kim, would you like to pause to see if there are any questions or comments from the audience?
Yeah, we encourage anybody that has any final comments or questions to go ahead and put them in the Q&A box.
Well, I can say, Shrag, that was an excellent summary. I really appreciate your putting this together in a synopsis for us all with your slides. I think you encapsulated a lot of the discussions and all the 4 theme areas. And I also want to, before I start the next phase of the conference, want to thank Dr. McAllister, Dr. Mechanic, as well as Carrie for all the work they did for organizing this conference, and all of the speakers, as well as the panel members. I think that the strategy of this working group, as well as the symposium, was extremely successful. I want to say that it took a lot to come together, but these themes are so important and they are cutting edge, and we need to come to a consensus as to how we can build this together and to work together to figure out a lot of the work in exposomics, not just for cancer, but also other disease areas too, and health. And what are those exposomics around normal health as well? So with that, I really felt like this was a conference that was not only knowledge sharing, but ideas generating. And that's very important as we think about how this will serve as the platform for other symposia for the NCI, for the NIEHS. And I'm really glad to know that there's a follow-up symposium on June 29th to the 30th. So thanks again to all of you for organizing this. And I hope it's one of many symposia that will lead to guidelines and new program project grants that can be put together around this, as well as new consortia. Because clearly we heard very loud and clear, and it was echoed throughout the conference, that increased sample size, more detailed data, generating that harmonization between data is very important for all of us to be able to make those new discoveries for a sustained longitudinal period with certain exposure time periods in mind. So a lot of these items were discussed today. I'd like to bring to the audience some questions that I think are imperative to try to get your feedback on, since your feedback is critical to how the NIEHS and the NCI can frame their solicitations of grants, or can frame future research for us as academics, but also for the world to make an impact. So my first question to the audience is, what study design issues should be considered for cancer molecular epidemiological studies incorporating environmental exposure measures and other omics data? What study design issues do you think need to be imperative and top of line? We talked a lot about case control studies, but are there others that you would suggest we make sure we can prioritize? And what issues might there be in those study designs. Anyone have some thoughts, or I'm wondering if the participants, do they want to use the chat, or does anyone?
I see a few people have raised hands.
Okay, great. I cannot see those people. Do you want to call them out? Sorry.
It looks like Francesca, Luca,
Okay.
and Matthew Duvall and Nat Rothman.
Great. So Francesca, Matthew, and then Matt. Go ahead, Francesca. Thank you.
Hi, thank you. So I just wanted to bring back into the conversation some of the topics that we touched on in the last section of these 2 days in terms of study design and experimental approaches. So I think, the in vitro exposures and the omics assay from cellular model, and we talked also about organoids, are a nice complement to the epidemiological or the case control studies. So we mentioned using them potentially as a follow-up for in vitro validation. And there's of course some challenges there that we briefly discussed in terms of defining timing of exposure and dosage of exposure that are biologically meaningful, but even taking into consideration that some simplification are necessary to be able to perform these studies, they will still, in my opinion, I think from what came out of the panel as well, be essential to bring a validation to large GWAS or case control studies.
Great, thank you. Matthew, Matt, and then Nathaniel.
Yeah, I think one of the things that I'm really interested in is determining how we will establish kind of power for these much more complicated studies, not just in terms of our main analyses, but also, there's a lot of these exposure studies that are that we do that are highly complicated. A lot of exposures are not just uniform across society. We have racial effects, we have gender effects on averages, population averages for who consumes this or consumes that. I think trying to come together as a community and figure out how to address such a complicated issue as the environmental exposure is really important.
Agreed. Matt.
I actually think it's relatively straightforward.
Yeah, thank you.
Nat, am I next?
Okay.
I think it's actually relatively straightforward. First, Ricky Peters had suggested or queried the meeting, is it possible to put together an environmental atlas, so to speak. I think that would be something that's really cool and tractable. As I had mentioned before in my comments, if group of people could decide what are the key exposures, and here I'm talking about like environmental exposures, not lifestyle, not diet, not alcohol, not tobacco, though those are good models. I'm talking about the things that industry generally pollutes our environment with, though sometimes pollutants, toxic compounds are present naturally as well, like aflatoxin and arsenic, or aristolochic acid, or things like that. I think that if we decided what are the exposures that we really need to collect more information about, that we could put together a series of either cross-sectional studies or short-term longitudinal studies with populations that are optimized based upon their exposure as far as levels and wide range of exposure. Geographic diversity, racial and ethnic diversity is really critical, because there may be interaction between not between many characteristics of race and ethnicity that could impact the exogenous exposure biologic effect and ultimately disease relationship. And that I think these kinds of studies, and the question was molecular epi, so that's epidemiology. These kinds of studies could be done with not just identifying the right populations and through an international effort, but really putting together semi-standardized protocols, first as far as exposure assessment, which is absolutely critical, often lacking in either in detail or lacking in enough effort in studies. So exposure assessment is absolutely critical. In epidemiology, the classic teaching is 50% of your effort should go into exposure assessment. That still holds. Ultimately, biologic markers are not necessarily a panacea to really well conducted exposure assessment, which is well beyond just asking people what they're exposed to. Obviously, the environmental epidemiologists understand what the state-of-the-art methods are to try to do that, and I won't go into those. But the idea that we could study a series of populations exposed to agents of concern, and then with standardized sample collection protocols and standardized analysis protocols could begin probing into what are the biological perturbations that these cause. We could put together a library of what these exposures result in that could be used to interpret what will be a tsunami of data that we will experience as these technologies are inevitably already being applied to the many prospective cohorts in the world to help in trying to interpret what comes out. So I think it's tractable. I think it's relatively straightforward from a conceptual perspective. And I think it would be really cool if some effort could be put into trying to not necessarily centralize, but to try to at least bring people who are doing these kinds of studies together so they could try to communicate across studies, across exposures, across study designs, and maybe develop a bit of a consensus to try to move the field forward.
Yes, thank you, Nat. That's great. I see, so Catherine's next, and then I see some other people that have joined to answer that question. There's also one from the Q&A for anonymous attendee about synthetic cohorts of different age groups to capture exposome and phenome throughout the life course and clever longitudinal studies. Those are good points. I don't think, I mean, of course, we want to incorporate that too. So don't think we have any specific answers yet, but that's a great idea. All right, Catherine, you're next. Thank you, everyone.
Yeah, I guess my question was on a practical aspect as we're trying to put those studies together is always to go back to the sample size consideration, because now we have a really complex question, complex design, heterogeneity on both sides, the disease, the exposure, multi-layers. Ultimately, we all, put studies together and need to have some guidance about what are we talking in terms of sample size? Are we still within the traditional framework of sample size consideration? It seems to me that I'm always struggling with this, and I'm still being assessed with that. And I don't think there's a consensus on what's right, what's wrong. So what do we do?
Yeah, you're absolutely right, Catherine, that with multi-omics, it blows it out of the water, right? With all the exposomics and then understanding computer learning algorithms, understanding how to correct for multiple hypothesis testing, which is essential. And that's what Chirag mentioned too. So I think I don't have any answers. I don't know if Chirag, if you do, but these are excellent questions as we think about the different tools we have for computational analysis. And then what do you need to do to begin to get there to test hypotheses? Any comments, Chirag, that you have on Catherine's comment?
No, I lean on the experts here that could take it, but I totally sympathize that. I think along with our measurement modalities, just like when the GWAS came to fore, the very first papers that were written is how many genetic variants are we looking at, and what are the proper thresholds by which we need to be correcting that. I think the same things need to be done once I think we harmonize or come to a consensus about what tools we are using to measure.
Yeah, thank you. Sorry,
I can probably provide, I guess, a little bit of how I think about it. So,
right,
as you point out, there's the multi-dimensionality kind of aspect. So there's always a need to correct for multiple tests, and that, of course, erodes power. The alternative is we try to synthesize up front and do dimension reduction kinds of things and build models. And the payoff there is we have less comparisons down the line, but the the negative trade-off is we're giving up some information and we're kind of bound by whatever the R squared is for the model that we've developed, an expression QTL model or PRS model or some other kind of model. So, ultimately, I think when I think about sample size and power, really hinges on a well-defined hypothesis. And so, when I sit with my colleagues to think about this, yes, we can collect all of this data, but when you boil it down, you've got to have some clear hypotheses on which you can even think about what power and sample size mean. And so we talk about, well, how many tests are you going to be doing? If you're going to be using some exterior resources to build a model, what's the R squared for that model? And once we kind of know those building blocks, and it's relatively simple actually at that point to kind of come up with a realistic power calculation in a given design with a given sample size. So I guess, to kind of boil it down, I guess where I see the emphasis needs to be placed is in building as good of models as we can to try and get those R squareds as high as we can when we're going to be using kind of dimension reduction. And this gets into things about which population we're looking at and making sure that whatever model we come up is really going to apply to all of the various subgroups that we may be analyzing in a particular study.
That's great, Jim. I actually was going to ask a question about methods and computational approaches. So we're already sort of on to the next question. But before we sort of close that, I think that Leah also had her hand up, and then Ivana and David. I didn't know if you wanted to comment on anything.
Yeah, I had my hand up. I wanted to point towards a question that Dean Jones put in the chat and it was directed to Shrug, but I think it's for everyone, is what is the path forward? And so I'm interested to hear, we heard some thoughts already about paths forward, but I'm interested to hear from the group, from all of our panelists, what do they think the path forward is?
Well, just one thing on the last one, on the power considerations is, I hope that as a person who's not writing external grants anymore, I hope it's useful in study sections now if people start considering their power being what they can build on. I think the last thing that Dr. Gauderman said was, what things you're going to reference and build upon. To me, that says that your power can be increased if you can connect up with resources already, things like ELK, other things that people are talking about to harmonize these things. I think that makes a given study quite a lot different if it's designed from the beginning to build on something and to add to it. And so, I hope people direct some of their efforts toward resources from EPA and elsewhere and NIH about, the chemicals dashboard, CTD, other big things like that, that you can effectively leverage the power of any individual study with some data that with the proper meta-analysis can hopefully augment that.
Thank you, David. That's great. Yeah, I think, Leah, it's a great question to all of our participants, sort of what is the path forward? Would you like me to ask that at the end, or you want them to try to give you feedback right now before we talk about the other questions? What would you and Kim prefer?
I was thinking, I think it was kind of a logical flow from some of the conversation, so we could talk about it now, and
Great.
then, yeah.
Any other comments on, where do we take this? Where is the best path forward now as we think about research and exposomics around cancer incidence and prevalence?
We did mention briefly that there is this multi-omics consortium that's starting to form, and of course there's TopMed and a lot of different efforts at the different NIH institutes in coming years that are going to try to tackle this question of coming up with standards and best practices. So if folks have comments on critical issues or concerns, or what we should really be paying, NIH should be paying attention to with respect to that, that would be wonderful.
Thanks, Kim. Stephen, do you have a comment?
Yeah, so, I don't think there's any one specific path forward. I think most of us would probably agree with that. There are definitely, exciting areas where we're seeing large scale data generation that's being harmonized in different places. You mentioned Top Med is one example. All of Us is another, where there will be, multi-omics over time and, increased data generation about environmental exposures, may uncover some relationships over time, although, it still gets really hard to discover these in large scale, multi-dimensional human cohorts. So, I think continuing to focus on, how to get people data is going to be, where you see the biggest dividends. And another project that I'll mention is there's a common fund effort called MotorPack, which is looking at exercise as an intervention and studying molecular omics after that perturbation in a few thousand individuals. And so while people might not be specifically as interested in exercise for everything, I think these types of data can act as a model for thinking about when we do stuff to people, what happens at the molecular level, what types of changes are present, what are the dynamics of those changes, both in an acute period of time, but then also over a longer period of time. I think from those types of studies, we might learn a little bit more about how different omics inform, different types of exposures and activities.
Great, thank you. I think we touched upon, Stephen, the also what you bring up, which is what are those resources? What can we use for data sharing, data gathering? That was such a common theme in this, as well as the Atlas approach. And I think that if people are interested, the CZI, the Chan Zuckerberg Initiative with the BioHub, there are some publications that Carrie also showed in terms of transcriptomics cell atlases that are being done on the healthy control level that can serve also as examples and templates for all of us as resources. And to that point, are there any other resources that in particular might be useful for performing multi-omics integration? And I leave that for the audience. Feel free to just answer in the chat box too, because Stephen has mentioned some of them, but if there are others that we can share, Kim had a bunch of resources that are available in the NIEHS website and others, and I think if we could all share those, that would be helpful to our science. Are there any other key resources people think are going to be important, or resources that you think will be important to generate to ensure excellent and rigorous research?
I'll quickly throw out another one too.
Yeah, thank you.
So in terms of what the NHGRI is doing around the IGVF consortium, so this is looking at genomes, interpreting genomes and understanding variant function. It looks like a lot of projects are actually, looking at perturbations at a molecular level to understand not only, what are genetic effects at baseline, but under that perturbed state. And so, it could be that there is interesting opportunity to look at those types of model systems and the high throughput assays, which are exposing environment specific or context specific genetic effects, and translating those up into higher level epidemiological studies.
Yeah, thank you, Steve. I noticed that Carrie in the chat was able to put in a link to the NIEHS data standards RFA that will be due relatively soon. So that's also great too for the harmonization aspects that Dr. Kraft mentioned and others mentioned yesterday. That's due in May. Thank you. And then I noticed there's some Q&As. Interestingly enough, Dr. Gauderman, if you don't mind taking this one, do exposome studies by definition negate the ability to form a hypothesis?
No, I don't think so. But ultimately, as I said, any power sample size calculation is going to depend on having some hypothesis that you are interested in. If the goal of this study is to kind of do dimension reduction and build models, then we're going to be thinking more about, AUC and sensitivity, specificity, R-squareds, those kinds of things. But if ultimately the goal is to look at a, exposure versus outcome, looping in some of the exposome data, then As I mentioned, it's a matter of sitting down and seeing what are the testable hypotheses that we can pull out of that. And that's then a trade-off between how many tests are going to be done versus how much dimension reduction is going to be done, and how well we can estimate what that dimension reduction is going to look like.
Agree. Yes, I agree. You can always test the hypotheses, and through exposomics get more data to disprove or prove the hypothesis. So, and I just noticed that Chirag also linked for us the NHGRA's Anvil project for more resources for all of our participants and panelists. And then finally, I think there was another comment from a participant about integrating additional modalities like GIS, like geospatial data. And I think that brings me to the last question, which is, as we think about what resources are available, what environmental outputs are important for us to integrate, right? There might be some, like Nat was saying, that aren't so important. Let's prioritize those that we're most worried about, given the data that we have now, and we can do that carefully and thoughtfully. But with that framework, given the fact that we want to begin with the end in mind, we want a solutions-facing type research with some policy-oriented outcomes, that certainly has helped with many cancer-causing agents that now have been reduced via regulatory guidelines. How can we best use the Omics data set to support cancer hazard evaluation so that we can help our colleagues at the FDA or the EPA or the WHO or other institutes to be able to best frame some regulations around these potential environmental toxic exposures, as well as dose response assessment? I'll leave that as my last question to all of you, because that is when the rubber hits the road. How can our science help policy and implement and protect the public?
Carrie, David probably has some answer to that, but I know Dean has his hand up.
Okay, Dean,
yeah.
thank you.
I was going to completely change the subject, so go ahead, please go ahead.
right,
well, real
thank you.
briefly, I would say, my quickest answer to that would be that anything that's a new type of data that can be called omics, it's about the reliability and the repeatability. And so I'm going to stop short of using the validation word, because that's a particular regulatory process. But repeatability and reliability are key. Reproducibility, whatever, they're nuanced in those statistical definitions there between those things. But I think with once it can be repeated and what you're going to get, it doesn't have to be the exact same. It's not a tolerance interval. It's not that kind of word. It's that, is this a consistent marker of something? Can you tie it? I think that's going to jumpstart any sort of approval or regulatory use process. That
you, David. Okay, Dean, different subject.
Oh, well, that was really a wonderful discussion there. And I think, that's actually gets the point I would just add to that is, in terms of all of our analyses, we do have to deal with exposures that hit a very small fraction of the population. And this is the concept that, in early genomic studies, they were talking about non-replicability. But the truth is, if you have a population that is exposed to something and that population isn't replicated elsewhere, you will not be able to replicate those results. And that poses a real problem when you get to precision medicine. But that's not what I was going to talk about. What I wanted to jump back to was health inequities. That this is an extremely challenging issue. And I would just like the panel to now, as we've gone through everything, to come back to that question: is how can we actually bring that as a very important component of what we're doing?
Absolutely, Dean. I think Nat might have some comments. I certainly do. The environmental determinants of health are coming together from the WHO as well as other institutions. So I think we need to be there, just like social determinants of health, and make sure that we prioritize those. But I wanted to hear from Nat. Thank you.
I wanted to thank you, Carrie. I want to address the query that you posited to the group, which is how can this work contribute to basically the risk characterization, risk assessment process that organizations like the EPA does and WHO through IARC, if we're talking about cancer. So this has nothing to do with precision medicine. This has to do with basically how can this work contribute to impacting public health applications that could result in regulation, either banning of chemicals or reducing substantially the levels of things that are in the environment or that are manufactured and end up in the environment. And what I just wanted to briefly note is IARC has International Agency for Research on Cancer has developed a more systematic approach to looking at the biological function of compounds that it's considering an evaluation of carcinogenicity. And based upon some work that Martin Smith has done at Berkeley, developed this idea of the 10 key characteristics, where they sort of define, sort of going backwards of the established carcinogens, what were their, and what are those their characteristics, and came up with biological functions that you might look for to decide, is there enough evidence to support the biological plausibility. Many of these types of assays are what we might call them, from the genome, using the genomic paradigm, candidate biomarkers. So they have a tendency not to be omic biomarkers. And IARC is having a meeting later this summer. And one question is, what is the role of omics in this process? And there have been some interesting discussions about this. And even though on one hand it's early, on the other hand, there's the hope and expectation that as more work is done applying some of this kind of work, particularly I think metabolomics, transcriptomics, epigenetics, epigenomics, and we may be able to interpret more biologically what these endpoints mean, and are they ultimately consistent with contributing to an evaluation of whether something is carcinogenic or if it isn't. And again, I don't want to use the term validation either, but I think we're sort of dealing with this this huge amount of data, and exactly understanding what this means and translating this to biological meaning as far as different biological pathways and pathogenesis of cancer will be a challenge. But there is a lot of excitement that this may play, that the field of omics may have an important role to play, as agencies like IARC and the EPA, many agencies are getting very excited about this idea, can start looking at omic data and use that to contribute to ultimately a decision about risk characterization.
Thank you so much. I know we're coming to the bottom of the hour, and we'll be ending around 4:30 after some closing comments. But I do want to close. There's one more comment in the Q&A about potentially having a specific study section on multi-omics and exposomics. And I would love to get all of your feedback because it sounds like that would be very enabling, and I'll be the first to volunteer for that study section to be a part of that if you'd have me. So what do other people think? Carmen?
Sure. I think that's great, Carrie. I mean, because I think one of the challenges here is a lot of this is hypothesis generating, right? And when that is reviewed, it often is not reviewed well, because we're coming in, we want to do discovery, we need to do discovery. I think for a lot of the, I mean, a lot of environmental health in general has to be discovery, because it's new chemicals are coming along and we don't know what they do.
Right.
And so it really has to be about discovery. But that's not something that's valued. It seems very much like that's something you have to really argue for. And I think we end up losing a lot. And I think we get a lot of biased results because we have to drive toward this, more, centralized idea, instead of like trying to think more broadly and trying to really understand what's going on. So anything you could do to get people to open up to the idea of doing more discovery science, I think would be really helpful.
Yeah, great. Well, that's excellent. I think on that note, well put, Carmen. I'll hand the baton over to Kim and Leah. Again, I really appreciate everyone's attention and thank you for staying on until the very end, because this has been a very fruitful, very engaging discussion. So thank you, and thank you for the meeting organizers as well.
Great.
Okay.
Thank you, Dr. Naidu, and thank you, Dr. Patel. That was really a great summary and a final panel discussion. I want to thank everyone for joining us today, and particularly thank our speakers, panelists, and moderators, and all the work you did to pull this all together. NCI and NIEHS will be using the information that you've provided to think about our paths forward of how we can advance this field. I want to also thank Carrie Volker and her team at MDB for all their work behind the scenes to make this meeting seamless. And I want to thank Alex Hudson, our PMF fellow, who did a lot of the legwork helping us pull this agenda together. Note that we're going to be posting a recording on the website in the next several weeks of this meeting, and we'll be working to develop a report that will also be posted. And with that, I want to thank everyone for joining us today. Have a great rest of your week. We really appreciate all that you've done. So thank you. And Kim, do you want to say thanks as well?
Yes, thank you very much to everybody that participated. It was very helpful.
Great. Thank you.
Thank you. Bye, everyone.
All right.
Bye.
Have a good evening.